All right. So today is part 2, gene regulation part 2. So today we're focusing on eukaryotic organisms as opposed to prokaryotes, which we talked about last time. And so I got my timer set for 1 hour and 15 minutes because I went a little over last time, but I'm surprised it didn't take me 3 hours because it's a lot of info, and now I can kind of keep track. But anyways, so today we're focusing on eukaryotes. Now, just a quick sort of refresher. Who can tell me what defines a eukaryotic organism, a eukaryotic cell, as opposed to a prokaryotic cell? Of course, there's no one here, but if you're answering it, in your own head or on paper or out loud to your dog, whatever it is, that's kind of the goal. Try to be as interactive as possible. So a eukaryotic cell and eukaryotic organisms that are composed of eukaryotic cells, if they're multicellular, there are unicellular eukaryotes, so don't forget that, like amoebas, certain parasitic protozoans as well, phytoplankton. Anyways, I digress. A eukaryotic cell is complex, typically larger cells than prokaryotic cells. They contain membrane-bound organelles, such as the nucleus, which houses the genetic information, our DNA, mitochondria, lysosomes, Golgi apparatus, endoplasmic reticulum, etc. So today we're going to be discussing how genes are regulated in eukaryotes, and we're going to see that there's some similar trends, similar themes with the prokaryotic cells, with transcription and all this stuff, but there's some nuances and some differences that allow for more regulation, really, and more fine-tuning and tweaking. And we sort of started our discussion last time with sort of talking about how different cell types arise from a single cell. So as we know, all life starts as one cell. And then through successive mitotic divisions will give rise to a multicellular life form. Or if it's a single-celled organism, it can clone itself through asexual reproduction to make two separate organisms. So, okay, sorry, my distracting thing, text, but man, if I do that, do not disturb. Okay, let's see if that works. Okay. So where was I going with this? Okay, starting with a single cell. Now, the key here is that the only way that this single cell can give rise to a variety of different cell types, from skin cells to liver cells to brain cells or neurons, same thing, is through gene regulation. Which genes are activated, which are inhibited. And today we're going to explore how this happens in us, eukaryotes. last time we saw kind of gene regulation in prokaryotes in response to, the presence of tryptophan or lack thereof, to lactose availability. So certain signals can also be integrated and manipulate gene activation or protein production this way. This is called signal transduction. So it's the sort of chapters they kind of, are highly related, right? But today, yes, we're gonna really look at how the cell does this in eukaryotes, and how we get these different structures and thus different functions in these cell types, from the development of one cell to billions of cells. So first, let's do a little quick review of sorts now. Don't focus yet. This will be a good picture to come back to later, but I want to just kind of take a quick overview of our central dogma of cell molecular biology. So as we've discussed, the central dogma is this sort of flow of genetic information from DNA to RNA to protein. It's so critical in all cells, all life forms, that it's called the central dogma. It's just this massive theme here. Okay. So, let's see. With this, I want you to focus on DNA, right? Being transcribed to produce a messenger RNA molecule, single-stranded, and then the RNA just kind of really, again, this is a eukaryotic cell. So let me backtrack for a second. In prokaryotes, we saw, well, we have this exact sort of mechanism going on where DNA is going to be transcribed to produce messenger RNA, which will then be translated at a ribosome into protein. But we know that there's no nucleus in prokaryotes, no nucleus. So the DNA is kind of just floating in the cytoplasm. It's free floating. It's all of this stuff happens in one space. But here, no, we've got different areas of the cell where things occur. And for eukaryotes, the nucleus, we get transcription because that's where the DNA is. And so we produce the mRNA. The mRNA is then has to be transported to the cytoplasm through the nuclear membrane. There are little pores in the nuclear membrane that the mRNA can go through. It's small enough to go through. And so mRNA in cytoplasms. Now that we're at the cytoplasm, translation can occur because this is where our ribosomes are. And we know that, and we learned that mRNA being translated into protein or a polypeptide chain occurs here at the ribosome. And that's sort of what I want you to focus on right now. So the the key here is that there's a lot more, as you can even see with all the degradation of mRNA, RNA processing, protein. There's a lot more opportunities for eukaryotic cells to regulate protein activity. Whether proteins are produced at all from RNA, whether they're active, how long they're active. This is going to really be important in how cells specialize in structure and function. What proteins are they making? How much are they making? How long are they sticking around? So we're going to learn, these are the mechanisms. This lecture is going to focus on the different mechanisms of eukaryotic gene regulation. So we're going to start with this general definition, differential gene expression. Okay. So differential gene expression okay, so this is the process by which cells become specialized in structure and function. Differential. Oh my gosh, it went all dark on me. That's annoying. Oh, it's because of the camera and the light. So let me do that. Okay. I think I get it now. It's kind of annoying. I see what it's doing. If it continues to bother me, I'll pause the recording and fix it. Probably set the brightness like at a set brightness. Okay. So differential gene expression, right? Different genes expressed in different cell types. Okay. So it's important to recall, and I think I mentioned this last time as well, that all of our somatic cells have identical DNA, identical genomes, genomic sequences of DNA. It's the same. So how does a cell differentiate into a keratin producing skin cell, structural supportive protein and protective cell, a myosin and actin producing muscle cell? These are the contractile proteins, the fibrous proteins responsible for movement in our muscle cells. An antibody producing white blood cell, a digestive enzyme producing pancreatic cell, etc. Now, they look different, they act different, they're functioning different, they're structurally different. So, but that's not to say that a muscle cell doesn't have the gene in its DNA, in its genome, that encodes for keratin that a skin cell expresses. It's not to say the muscle cell doesn't have digestive enzyme genes, but it's not making them. They're shut off. It's not a muscle cell's job to digest food, right? Okay. So let's take a look at an example that I like from the textbook, and kind of, well, just to visually sort of see how this plays out. Come on. Okay. So for this example, there's a liver cell and a lens cell. Now the lens cells are in the retina of the eye. So an eye cell basically, and a liver cell. So I like the top picture. And I like all of it really, but the top here is cool because it says DNA in both cells. So focus on that for a sec. DNA in both cells. Notice that they have identical DNA. They both have this DNA. They both have the enhancer, the promoter, the albumin gene, the crystalline gene. Now, albumin is a protein produced by liver cells. Crystalline is a protein produced by lens cells, but the liver cell has the crystalline gene. The lens cell has the albumin gene. They both. It's the same. So this will make more sense when we cover transcription factors and transcription activators in eukaryotes. But if you look at let's look at the liver cell first. The liver cell, we're in the nucleus here with the DNA, has these available activators, transcription activators. And then you look at the lens cell, it has different activators. So because these activators don't recognize the albumin gene, they're different. These are the activators that recognize the albumin gene. If you have these ones present, these activators, then you're going to see expression of this gene. You're going to have albumin produced. So it's all dependent on which transcription factors are available. Again, this will make a lot more sense when we talk about it, but I wanted to preface it with, it all depends on not the DNA sequence, it's the same, but it's the available transcription factors in the cell. So just keep that in mind when we get to it. So if those activators were in the liver cell, then you would see the crystalline gene expressed. So they're kind of calling the shots here. Okay. Let me see where I'm at. Okay. Oh, this is kind of nice. I can pause the recording to kind of review my notes, and because this is the first time I'm doing this lecture, both of these lectures, the first time. And I made them from scratch, so it's like trial and error, and I had to rearrange some things, but I think it'll work. But anyways, just keeping in the back of your mind about these transcription factors, okay? And they're dictating which genes are being expressed and which really aren't in different cell types. Okay. Now I want to take a look at a very cutting edge emerging field that is extremely popular right now and getting a lot of attention, called epigenetics. I would be remiss if I didn't talk about epigenetics in this lecture. And so it is something unique to eukaryotes, and eukaryotic cells is this field of epigenetics, and how epigenetics plays a role in gene regulation. Okay. So regulation of chromatin structure. This is the heart of epigenetics. And so I want you to recall what chromatin is. Now, During interphase, remember this is the cell cycle G1, SG2 before mitosis, right? Interphase is when our DNA and these proteins called histones form this complex that looks like spaghetti. It's not too condensed, right? It's like kind of stringy and loose. Well, this is good because it makes the DNA accessible for transcription, for RNA polymerase to come in and transcribe DNA to produce messenger RNA. Okay, great. So keeping that in mind, these latest findings have shown that there are actually two types of chromatin. Two types, one of which is a little more condensed than the other. One is a little more loose. So we'll start with the loosely arranged chromatin. So this is called euchromatin. Euchromatin. Gosh, can't with the light. Okay. Euchromatin is loosely arranged chromatin that is accessible by the proteins that carry out what? Transcription, right? And then, so I'm writing that. And then I'm going to write and its genes can be expressed. Maybe I figured it out. If the light's shining on the camera, it'll stay light like that. So that's euchromatin. Let's look at what we call heterochromatin. Heterochromatin is a little more compacted, a little more condensed. Now we learned that chromosomes are like highly condensed. If you zoom in on it, it's like super packed in, coiled, folded up. Just no transcription is going to happen at all. Like you can't get into the DNA and unwind it and read the sequence. No way. Heterochromatin is a little more dense. I forget if I have a picture of both of them, but essentially it's just the DNA is less accessible. You can't really carry out transcription in areas of heterochromatin. So heterochromatin is densely arranged chromatin that is less accessible for transcription. Transcription. Then I'm going to do a dash. Genes are generally not expressed. Okay. Okay, I just wanted to check. I paused it. I wanted to check if I had a good picture, and I like this picture better than the textbooks a little bit. It's a little, and so the one in the slides. Okay. So if you take a look here, here's our chromosome, right? Just like super coiled up and compact. This would be like heterochromatin, the inaccessible chromatin. Nucleosomes are the term for just one of these units of histone proteins with DNA wrapped around them. But you can see that DNA is wrapped around them, and then there's a string here. This would be like euchromatin. The DNA is much more, proteins can come in and carry out transcription here more easily, right? And so it zooms in. I like this picture because it keeps zooming in on a nucleosome. And you can see that there's these histone proteins with DNA wrapped around them. And the reason I like this is because we're going to talk about epigenetic modification, the first of which has to do with these histones. Make sure the timer's going. Okay. Okay, and I want you to pay attention to this N-terminal histone tail. All that's referring to is that these are proteins, and proteins have an N-terminus and a C-terminus at either end of the polypeptide chain. So remember, proteins are chains of amino acids, these polymers of amino acids that we sometimes call polypeptide chains. And there's an N-terminus. If you zoom in at the molecular level and you see the amino acids, you'll see even in the individual amino acid, it has an N-terminus and a C-terminus. Basically referring to an amino group at one end and a carboxylic acid C group at the other. So at the end of each protein chain, you've got an N-terminal chain or group, and then the C. So there are these N-terminal histone tails that protrude outward from the protein, this globular histone protein. And these tails can be modified. They can be acted on by enzymes. And it's going to actually affect the chromatin structure, whether it's loose euchromatin or condensed heterochromatin. So let's take a look at that. Okay, epigenetic regulation, histone modifications, and DNA methylation. Okay, so in all eukaryotic cells, all eukaryotic organisms, histones can be modified. And this is going to affect transcription of genes in the DNA. And like, as I said, this N-terminal tail sticks out from the nucleosome, which again was that structure of the histones and DNA in those units, right? So let's go ahead and write. So we're going to start with histone, what we call histone acetylation. It's the first type of chemical modification that can be made to these histones. I'll get a new piece of paper, histone acetylation. So histone acetylation is the addition of an acetyl group. It's like an organic chem term of what that is. So I'll put it in parentheses. An acetyl group is a group, it's going to be bound to other atoms in the molecule. So I'll denote that bond here. I'll show you in a moment. Then a carbon, oxygen, carbon with 3 hydrogens. So that's what an acetyl group would look like in a molecule. So it's the addition of an acetyl group by an enzyme, by a modifying enzyme. And you don't have to know the name, but one of which is called a histone acetyl transferase. And that's again, we don't like to name things like for no reason or randomly. It's transferring an acetyl group, right? So makes sense. By a modifying enzyme to an amino acid in a histone tail, and those tails that I just showed you. So it's going to acetylate a histone tail. This promotes transcription. By opening up chromatin structure, loosening it. Next sentence. So histone, so that's histone acetylation. It's going to really activate transcription in regions of chromatin that are more like heterochromatin and condensed. Oftentimes when you see chemical modifications, addition of chemical groups, things binding to one another, you're going to see a shape change with that. So yeah, I just like to mention that because it's just such a powerful common theme in biology. With all these chemical reactions, you're going to see a shape change in the end molecule. Okay. Next sentence. So histone methylation. So histones can be acetylated or methylated. Histone methylation. On the other hand, right? So I'll just do that. So I'm starting with histone methylation. Oh gosh, brightness. Histone methylation. This is the addition of a methyl group. Which is to the bound CH3. The rest of the molecule will, what we can say is downregulate or inhibit, prevent, decrease transcriptional activity, downregulate transcription. And if you guess why, it actually condenses the chromatin rather than opening it up. So histone methylation. Don't worry, I'm going to introduce some colors soon because it's too black and white right now. Okay. So we'll see, but I was planning to talk a little about cancer towards the end of the lecture. Hopefully we'll have time, but we'll see. So now we're talking about methylation. So going away from histones, let's write in blue. In addition to modifying histone proteins, we can actually modify the DNA itself, crazily enough. In addition to modifying histone proteins, the DNA itself can be modified. And the mechanism we're most familiar with is called DNA methylation. Again, methylating. So whenever you think of methylation, we're actually downregulating transcription. So DNA methylation on certain bases. Typically cytosine. I'll write that in as well. Cytosine can be methylated. Now, again, DNA of genes that are more heavily methylated are not expressed. So methylation really silences transcription, or silences genes, we call it. Gene silencing, we're turning them off. We're not going to carry out transcription or produce that protein product encoded by that gene. I think I'm gonna fix this because it's bothering me. Hold on. All right, the brightness won't change now. I don't know if you guys see that, but yeah, anyway. Okay, so I can just hold that up. Okay. All right, so like I said, this is a really popular field right now, epigenetics. And so I wanted to mention some relevance, the significance of these types of modifications to histones and DNA. If you notice, the DNA sequence didn't change, right? With methylating or acetylating the histones, we don't change the DNA sequence. And yet, we're regulating gene expression. We are going to see genes that are produced or not produced, activated, inactivated. We never used to think this was possible. We used to think that, as biologists, we used to think you had to affect the DNA sequence somehow. If you have a gene for insulin, if you if you mutate the DNA, you change the DNA sequence for that insulin gene, it's going to affect the protein product to be, it won't assume the correct structure, or it's just going to be defective in some way. It's not going to work. You're not going to get the insulin. But with epigenetics, you can methylate that gene and you won't see insulin produced. So you won't get functional insulin activity without, you might have a functional gene. You might have the normal allele, right? So importantly, epigenetic patterns are passed on to future generations of cells. And we call this epigenetic inheritance. So as the DNA replicates prior to cell division, the enzymes actually come in and methylate DNA before, and the histones as well with acetylation and things. They're good. The patterns are replicated before the cell divides into two, and you get the chromosomes and everything. It's pretty amazing. Okay, and then again, DNA mutations, they're typically permanent. Like if you change the sequence, if you mutate my DNA with ultraviolet radiation or carcinogens or inflammation, like these things, that's going to affect protein production permanently. You're taking the cookbook and you're erasing the recipe, or you're rewriting a different recipe that's not going to produce the meal or the whatever you're cooking. You're not going to get the soup or the pie or whatever. But epigenetic modifications are reversible, and they can respond to environmental changes. Methylations, acetylate, all these things are, there's demethylation, there's deacetylation. You can remove these groups, you can add them. It's flexible. Again, this is pretty new for us. And it does explain, we never really understood fully how identical twins can end up being so different later in life until they looked at epigenetic patterns, which is something you can develop during your lifetime. So one can develop schizophrenia or another type of disease, type 2 diabetes, obesity, etc. Depending on how they were raised. It's pretty amazing. So two genetically identical mice, okay? They have this coat color gene that's overexpressed during development. So the one on the left here, you can see that's what that looks like. Now, the right's mother looked like that. Okay, it had this overexpressed gene, but the right's mother got pregnant. Okay, and they fed while the offspring was developing. They fed the right's mother a diet rich in folic acid, which contains a lot of methyl groups. And so these methyl groups actually methylated the offspring's DNA, believe it or not. And that's the offspring. You would think that you'd get something it has this overexpressed gene, it has the same genetic sequence as the mother, this fur gene, that it would look like that. But no, the methylation actually downregulated this gene. It'll prevented it from being transcribed for too long. And so it only got the correct amount of coat color. And amazingly, this mouse got pregnant as well without even being fed the folic acid diet. And the mice of this mouse, the offspring of this mouse, had the same fur color and they didn't feed this one the folic acid diet, meaning this is epigenetic inheritance. The epigenome, the methylation pattern, was passed on to this mouse's offspring. Now, this example, a little more relevant to humans, blew my mind, and I'll show you why in a moment. But the Dutch hunger winter. So, part of why I love biology is because I like learning about a lot of different fields, and biology can be relevant to so many different things, from psychology to history to even philosophy. And here, this is a bit of history. The Dutch hunger winter. So towards the end of World War II, the Nazis were going to bring in more troops into the Netherlands, but the Dutch railroad workers boycotted the Nazis from bringing in more troops. So the Nazis, to retaliate, blocked food delivery. Assholes. And so 20,000 people actually died, Dutch people, of starvation. And there were pregnant women. The pregnant women who didn't die during this prolonged starvation period gave birth to their offspring, and recent research from 2018 found that offspring born during that period versus women who got pregnant shortly after or before and gave birth in the Netherlands, these adults, now today adults, the ones born during this starvation period had higher rates of obesity, type 2 diabetes, and schizophrenia. Statistically significant to show that enough women during the prolonged starvation, the higher rates of this. But they had the same mothers. So they looked at the same mothers as well, who had more children, but after this, 9 months, they were fed, they were healthy. Those children didn't experience this, but they had similar genetics, same mother. And when they looked at the methylation pattern and acetylation of the histones, they saw that it was very different among these siblings. And so this is revolutionary a bit, mind blowing. Why? Because, Lamarck. Remember we talked about Lamarck as being wrong? Evolutionary biologist who theorized that if a giraffe needs to reach fruit at the top of a tall tree, it could stretch its neck, develop these muscles, eventually getting to the fruit, and its offspring would inherit those muscles. But we know now that that's false, right? That characteristics acquired during an organism's lifetime are not passed on. Darwin was sort of the pioneer of showing that that's not the case. It's these random mutations which the environment then selects upon for those who are stronger. It has nothing to do with the will of the individual. Well, based on that, how the mother eats, the health of the mother actually affects certain we thought DNA hard-coded genetic type traits like schizophrenia and type 2 diabetes, all these kinds of things that we thought had a genetic component, but it really, a lot of it has to do with the lifestyle of the mother and passing on these epigenetic modifications. It's pretty amazing. We thought, if whatever the mother does, the DNA sequence doesn't change. You'd have to change the DNA sequence for them to get X, Y, or Z. Turns out you don't. Biology is constantly evolving. Ha ha ha.